should have some additional time at the end for questions if there are more questions then. So our three speakers, I'm just going to briefly introduce each of them now. Our first is Gail Fanucci. She's a professor of chemistry at the College of Liberal Arts and Sciences, and her work focuses on structure, function, dynamics, and conformational sampling of biomolecules, and so this includes looking at HIV-1 protease and its mechanisms for drug resistance. Carla Mavian is a postdoctoral associate in the College of Medicine's Department of Pathology, Immunology, and Laboratory Medicine. She works in the Salemi lab, which is interested in phylogenetic analysis and evolutionary patterns, including those related to HIV-1. And Dr. Jenny Yamamoto is professor in the College of Veterinary Medicine's Department of Infectious Diseases and Pathology. She's been working in the HIV-1 field since 1984 and is co-discoverer of feline immunodeficiency virus, which is the feline counterpart to HIV, and the inventor of the first commercial Hiv vaccine. So thanks so much to our speakers, and we'll start off with Dr. Fanucci.
Did you want me with mic?
Yes, yes, so there's a room mic, but yes, if you could wear this mic, that'd be great. And actually, I forgot my one housekeeping thing, which is if you haven't already, please do sign in on your way out. We're not going to keep track of you. That's just because we've given you food. We need to keep track of how many people are here. And then if you could, fill out one of the blue forms that just lets us know how you heard about this event, what kinds of events you'd like us to host in the future. So thank you.
It keeps going back, but at least we practiced beforehand. All right, so good afternoon, and as Hannah introduced me, I'm a professor in the chemistry department here at UF, and what I want to share with you today is some of the research methods and tools that we use to specifically look at a concept of conformational sampling in HIV-1 protease, and look at this as a mechanism to explain how drug resistance evolution is occurring. So overall, my group studies a variety of different projects. I just want to have one slide that kind of summarizes the things that we do in my research lab. So we're interested generally in macromolecular motion and how the motion of the biomolecules is related to the functional state in a normal condition as well as in a disease state. So we study proteins, and this is the story I'll tell you about today, but we're also interested in membrane proteins and lipids as well as RNA. So what is HIV protease? I first need to explain the enzyme that we study. So as the name implies, it's a protein that's an enzyme. The ACE means it's an enzyme. So it's a protein that cleaves other proteins. And so it's an essential protein necessary in the viral life cycle of HIV infection. And what's shown down here are EM images of an immature virus that's not infectious and then a virus that is infectious. And maybe in this gray here, you can see that the capsid protein is forming, and that's what this black is here, and it has the nucleic acid genome inside. So if you keep the variant in this state, it's not infectious. And the protease, this enzyme, its job is to cleave these proteins that are represented here as balls on a string. So if you imagine my pearl necklace, each of those structural proteins is folded but connected by an unstructured region. And this enzyme, the protease, needs to cleave it. Right? So if I ask you how can it do that, this is the crystal structure in a space-filling model, you say it has to move. Right? So we're interested in knowing how it moves for how it generates its function and also leads to drug resistance. So if I ask you how does something move, right? If I ask you how does a horse run, you would think about looking at it with your eyes and maybe imagining a motion picture. Right? And this is actually the first motion picture ever made of how a horse runs. But what is a motion picture? A motion picture is actually a sequence of static structures that we know how they're linked together. And X-ray crystallography as an experimental tool is really good at giving us an individual structure. But as this slide panel shows, if I were to ask you what percentage of the time does the horse have 1 foot on the ground, 2 feet on the ground, no feet on the ground, right? Oh, and this should say 0 feet, sorry, that says 3. You can see if you were to crystallize the horse in this state, none of its feet would be on the ground. So would that explain to you how it was running? Right? You wouldn't get a clear picture. In 6 of these slides, you'd have 2 feet on the ground. But we also might have substates where here the back feet are on the ground, here the front feet are on the ground, one front, one back foot, right? So crystallography gives us a really good indication of the individual states, but if we want to know how something moves, we need to know all of the states, and we need to know how they're connected. Well, the method that we utilize in my lab can actually tell us the percentage of some of these states, and that's what we call the fractional occupancy. And so, ideally, when we think about biological molecules who want to know how they move, we would love to have the analogous kind of moving, but we can't do this with our eyes, right? We can't look at through a microscope and look at a protein and see how it's actually moving, but there are folks who do computational work in silico that can give you a snapshot picture of how it moves. And this is work from our collaborator, Carla at Stony Brook. So it shows how the enzyme opens up and then how it closes back down. So how do we experimentally validate this, right? So we want to connect this idea of the horse moving and our computational picture of how does the protein move. Well, we use a method in my lab that's called double electron-electron resonance, and that's abbreviated DEAR. And so this fellow becomes our group mascot. And essentially what we're doing is very similar to the antlers of this deer. We want to measure the distance between two sites in our biological molecule. And we do that by putting a specific spin label. So my lab does all of the molecular biology, protein expression, purification, and chemical modifications to put spin labels at distinct positions. And then we do this DEAR spectroscopy to measure the strength of the dipole interaction between those electrons. And that's proportional to 1 over r cubed, so we get the distance. So why do we care about the distance? Well, if we have conformational states, just like those states I showed you in the horse, and we know in the closed state the distance should be 33 angstroms, in semi-open it should be 36, wide open we hypothesize greater than 40. If we can measure those experimentally, we then know the population of those states and the fractional occupancies. So this is just an example of what the raw data look like. We get an echo curve, we analyze this in terms of the distance profile, and then regenerate that as a series of populations of distances that correspond to our states. Now I'll point out, these two structures actually come from X-ray crystallography. So you can go into the protein database and see over 300 different crystal structures of protease, and you can find structures in the closed form and the semi-open. These two guys over here, you might say, well, they look awful lot like crystal structures, but these come from our molecular dynamics simulations. To date, we have no crystallographic experimental evidence for this. We have distances in our data consistent, the ones in black with these two conformations. So we're still trying to prove that these guys exist and actually use them in some drug discovery schemes. So why do we care, and how does this get back to this idea of drug resistance? So this conformational model, this funnel that I'm showing you here, the bottom tips, we think of these as the stability of each of the states that our system can sample. The height, where did my mouse go? The height of this barrier would be how difficult it is to move from one state to the other. And we know that when we add inhibitor or substrate, we shift to the closed state. That's the most common. And the most stable when inhibitor or substrate is bound. But what we're hypothesizing is that drug pressure selected mutations, they alter the stability of these states, which then can prevent or modulate the way inhibitors or substrates bind. So what is the data that we have that suggests that? So what this figure is showing here in red are all of the sites where primary mutations evolve drug resistance. And so if you've had a biochemistry class, you might know if substrate binds in the active site pocket, the FDA-approved inhibitors that are designed to target this enzyme also bind in that spot. They're competitive inhibitors. So when the virus replicates very quickly, its polymerase that replicates its genome does not have proofreading ability, so there are random mutations that occur. The random mutations in the genomic information may result in changes in amino acids. And these are, in red, the amino acids that occur first in response to a particular inhibitor. And they typically alter the shape of the active site, which impacts the way inhibitor binds, right? This is the lock and key hypothesis. If you change the shape of the lock, the key no longer fits. But substrate also binds there. And so if you change the shape for inhibitors not to bind, you're also impacting the way substrate binds. So it's been seen through taking blood samples of folks who've been infected with the virus and they've been sequenced and they're developing drug resistance, that there are secondary mutations that evolve that are outside the active site. They regain viral fitness and lead to multi-drug resistance. And we want to understand the role of these mutations that are on the periphery because they're not changing the shape of this. And we've hypothesized that what they do is they change this conformational landscape. Right? So it's a novel way to think of how drug resistance is evolving. And when I started this project, I started at UF in 2004, and Ben Dunn and Maureen Goodenow kind of recruited me to work on HIV protease. And Ben's lab has studied nalfinavir resistance. So if a patient is infected with HIV and they're given nalfinavir, this mutation, D30N, in the active site evolves first. And then 2 secondary mutations are seen to co-evolve. And they recover enzyme fitness, right? So the kinetic activity goes up, the viral loads go up. But then they also lead to cross-resistance to other inhibitors such as ritonavir and indinavir. And the question is why? They did crystallography and saw no systematic change in the structure. And so we said, well, let's see if the conformational sampling is different. And so these are the raw data we made. So in their lab, they made each of the single point mutants, the doubles, and then the triples, and did the biochemistry. Right? And they published this work in 2003. And I got to UF in 2004, so this biochemistry work was published before I arrived at UF. So we made each of those samples, did the EPR, collected the raw data, and then looked at these distance probes. And you can see from the wild type to some of these single mutants, this most probable distance is shifting between 36, which is semi-open, to closed. And when we get to the triple, we see an increase in this population of wide open and this curled tucked. And so we went through and did a bunch of analyses looking at correlations between kinetics, Kcat and closed, Km and closed. And what we found is roughly linear correlations. If you stabilize the closed state, you impact catalytic activity. And you needed about 60 or 70% of the semi-open to be catalytically active. We also found really strong correlations with inhibition if we plotted the percentage of the closed and open-like states. So how do we interpret this? So if we think of these 4 conformations that we see in our data, we group these 2 together into open-like, because to get these linear correlations, we had to group those 2 populations together. So what we've hypothesized is that in the wild-type enzyme, you have a conformational landscape where your semi-open is the most stable. It's there about 80% of the time. The closed state is easy to get to, and the open states are hard to get to. But drug pressure selective mutations, they kind of flip-flop the relative energies of these closed-like and open-like states. So now these open-like states are there at a higher percentage of the time, so the inhibitors don't bind as well. And so this is a hypothesis that we've been investigating with other darunavir-resistant constructs and other mutations that we have a few papers published on, but in 15 minutes I can't tell you all of that, but I'd be happy to talk about that. So if we come back to this motion of the horse running, I talked about how we can separate all of the individual states, and those states can tell us something about conformation. But what else we can do is we can overlay all of those states and then look at what regions are static and what regions are dynamic. And so we also have a spectroscopic tool that allows us to do that with biomolecules. And we can use NMR, nuclear magnetic resonance, to look at backbone dynamics. And so here I'm showing you data from our wild type. And then we have one mutation, this E35D, that we've shown disrupts salt bridges between this region, and then a protease with 5 mutations. And I'm not going into the background of this method, but if we have the blue, that means it's rigid. Red is more flexible. So if you just look at the colors of wild type, it's more blue, and as you introduce these mutations, they become more green. So what we know from the EPR work is that these guys sample the curl tuck conformation, and they're more dynamic. Right? So we were asked in preparing these talks to relate the work that we do to a translational impact. So I'm a biophysical chemist, so a lot of what we do doesn't always have immediate impact in translational medicine, but what would be the impact of this work? So dynamics of protein inhibitor interactions are important for drug design. So the thermodynamic interactions between a small molecule and a protein, you need to take into account both the enthalpy and entropy. So you need to know the dynamics. So although the structures of B and E35 are very similar, the dynamics are different. So the inhibitors that bind normally to subtype B, the wild type, don't have the same thermodynamic binding parameters to these proteins that don't have drug resistance, but they have altered conformational sampling. And this mutation is particularly prevalent in Brazil and in Asia. So it indicates that perhaps drug resistance may evolve faster, and we may want to design inhibitors that better target these more dynamic enzymes as starting places for next generation inhibitors for other places around the world. And then if we think about the conformational selection scheme that I told you about, we have crystal structures for these two states. We don't have any structures for these states. And our data are indicating that every drug-resistant construct that we've studied always has a stabilized open or curl tuck conformation. And so what we're trying to do now is get some structures of these two states, right? Can we target these two states as inhibitor design? And the approach that we're using is we're using phage display to try and find antibody fragments or peptide fragments that will specifically bind in this open-like conformation and stabilize it so we can crystallize it. And if we then have the crystal structure, we can work with organic chemists to design novel inhibitors. And we are seeing that for multi-drug resistant constructs, these two states are stabilized, where this is the most stabilized state in the wild type enzyme. So I think I finished perfectly on time. So thank you. I'd be happy to answer any questions. And the funding for this work comes from both NIH and NSF. And here's a picture of my group from last summer. We need a new one for this summer. But thank you.
I have a silly question. You mentioned Brazil and some other place. How does the geographic entity influence this? Is it genetic population that the gene pool that particular population has?
So there's, and obviously in 15 minutes I have slides from other talks. So the subtypes of virus that are found throughout the world have natural variation in their sequence due to genetic drift. So the same kind of errors that happen in replication of the viral genome in response to drug pressures happen without drug pressure selection in other places of the country. So those lead to different recombinating circulating forms and subtypes. Right? And so if we look at the sequence of protease, and I swear we didn't plant this question, if we look at the sequence of HIV protease in different parts of the world, subtype C is what's in Africa, AE is in Asia, and then PR5 is a combination of mutations that are in Brazil and in subtype C. And you see that these are all of these distal mutations. They're not in the active site pocket. So the wording that we use in these natural polymorphisms that occur elsewhere in the world are secondary drug pressure mutations that we see in subtype B. Right? So the concern in the hypothesis is that drug resistance will evolve faster and in different ways elsewhere. So we did this work with Ben Dunn, and we looked at subtype C and AE, and if we looked at conformational sampling of subtype C, it had a much higher proportion of this wide open state compared to subtype B. And then the AE and PR5 had these curl-tucked conformations. And so that kind of got us asking the question, as chemists, what hotspots in the protein are responsible for this? Because if we can tell where in the protein and what structural things are changing in the protein, that gives organic chemists targets to say, I need to target there. I should think about how to prevent this from happening. And we just happen to be developing this biophysical tool, the DEER spectroscopy, that we use in a variety of different systems and started to apply it to this project. Yeah.
Sorry, I know there's not much time, but I did want to ask, you mentioned that you developed this very novel tool looking at examining the mechanics behind drug resistance. Is HIV the only drug, disease and obviously drug treatment that you're looking at, or would you find would this kind of methodology work for other drugs for other diseases?
That's a really interesting question, and when I've given this talk at universities where there are people from like Eli Lilly, and they were really interested in thinking about this, because when people think about drug resistance, and I have slides on here that summarize some review articles, they say, oh, well, there's a structural region, the structural reason. The first mutation changes the active site. You have a compensatory that may change allostery, the way a cofactor or inhibitor binds. And then they say, oh, or it's dynamics, right? And so the conformational selection is not the same as dynamics, but what we're seeing is there's an interplay, because as we shift equilibrium, there is more dynamics as well. We have not applied this to other systems, but it could be applied to them. One of the criteria, obviously, is a three-state system, because if you only have a protein that samples two conformations, you can't be flip-flopping, right? So it's unknown how many other proteins or systems would follow this, but yeah. Well, thank you.
Do you hear me? No.
Yeah, I can hear you. Let me just switch. Keep switching back. I'm not sure why. Where's my mouse? Yeah, it's just here. There we go.
Okay. Hello, I'm a postdoctoral associate in the laboratory of Mucosal in the Department of Pathology, and I'm going to give you an overview on what has been our research so far in the field of HIV pathogenesis and evolution, applying phyloanatomy analysis and more recently brain transcriptomics in a model that is the rhesus macaque model. Okay. So I'm sure you have seen this slide over and over. I just wanted to say that HIV pandemic still doesn't have a cure or vaccine and has been a very devastating disease, not only at the individual level but also for a lot of societies, especially in the developing countries where the largest population of HIV infected people is located. And even if the antiretroviral therapy has, as you can see in the blue line, has lowered the amount of AIDS-related death, as you can see, as compared to a green line that will be a scenario of AIDS without antiretroviral therapy. Antiretroviral therapy still is a long life treatment that requires a huge economic burden, and also, as we have seen previously, it has a risk of drug resistance, but also other severe side effects. As for example, neuro-AIDS, there is a conference varieties of HIV-associated neurocognitive disorders. And as you can see here in this graph, I don't know where the mouse is. Okay. In the post-therapy era, the HIV-associated dementia has been decreasing, while still the prevalence of milder and symptomatic forms of neurocognitive disorders are increasing, because mostly because the life of HIV-infected individuals has been prolonged, and so it's easier for milder and asymptomatic forms to appear. Neuro-AIDS is not only neurocognitive disorders, but also other conditions that are associated with HIV infection, as HIV-associated encephalitis. And HIV-associated encephalitis has been linked to early viral entry of the virus in the central nervous system, and also to highly and rapid viral evolution and diversity. And in particular, bone marrow monocytes are being linked to viral entry in the brain and the onset of HIV-associated encephalitis. So in my lab, we wanted to sort the role of the migration of bone marrow monocytes in the development of the HIV-associated encephalitis. And to do so, we have used an animal model that is very well established, that is the rhesus macaque. These macaques are naturally progressing to simian AIDS because we don't use any therapy, and they are infected with SIV MAC251. We longitudinal sample during infection different cell population and tissues, including bone marrow and post-mortem brain and meninges, and we analyze a region of the SIV genome that is GP120. We analyze this region by applying a single genome sequencing, and to the
the sequences we apply basic phylogenetics, but also Bayesian phyloanatomy analysis to study the viral evolution. So, Bayesian phylogeography has been forefront in the epidemiological studies because it allows a reconstruction not only of the history of the evolution, but also of the dissemination, spatial dissemination of a pathogen during an outbreak, for example. And this can be applied also in the intra-host level to study the evolution of the virus in an infected host. In fact, the infected host can be seen as an epidemic in a micro scale. And applying Bayesian phyloanatomy, we combine spatial and temporal inference to reconstruct the intra-host evolution of the virus, to understand the role of the evolution into the immunopathogenic processes, and to understand key factors in the pathogenesis of the virus. For example, these trees have been obtained applying basic phylogenetics techniques, and represents different macars and with sequences that derive from different tissues and cells. And what you can see here is that the virus is subcompartmentalized in a specific region of the brain. This means that the virus has entered in the brain later in the infection and has been evolving under negative pressure. That means there are specific genetic features that allow the virus to enter in the brain and obviously cross the blood-brain barrier and enter in the brain, replicate and adapt in the brain cells. But if we use phyloanatomy, Bayesian phyloanatomy, we can also not only understand which cells of type of cells have been infected by the virus, but also when and from which cell type the virus is coming from. For example, here we applied the Bayesian methods and we obtained posterior probability distribution for these ancestral node locations. This means that each of these nodes takes in account of the uncertainty of the tree, because the probability of the distribution that is depicted here in each node is showing you a consensus of all the possible likely trees that we could have in this infection. And in fact, what you can see here is that this ancestral node is represented 47% by sequences that are coming likely from bone marrow, but also for 43% of sequences that are coming from plasma. If we would have used basic phylogenetic analysis, we only would have one value at this point, and more likely that value would have been bone and marrow. This can be, also this tree, as you can see, is scaled in time, so that we can understand the seeding of the virus, not only in each ancestral node, but also in time of the infection, to understand which type of cell was infecting the brain or other tissues in that precise moment. And to have a view of the spatial temporal distribution of the viral infection, this is like a plot would look like. We have the distribution of the different tissues over time, and what it was very interesting from these macars that were infected with HIV is that the bone marrow monocytes are always the last that is contributing to the viral infection in the brain. And this phyloanatomy technique can also be applied for other types of questions that we might have. And actually the next question we have is how and which type of cells are involved in the viral rebound after therapy interruption. In fact, right now therapy, the new era of therapy is probably the structural therapy interruption, because there are several studies that have shown that if we have a therapy introduction over time, we will have a delayed rebound of the virus. The other topic that I wanted to talk to you about is the gene expression and the pathways that are expressed in the brain to identify possible features associated with neurocognitive disorders. To do so, we compared sequences from the frontal lobe of macaques with detectable viral load in the brain. That doesn't mean necessarily that the brain was infected. That means only that we detect virus in the brain. And we compared these sequences from the brain of macaques with no viral load in the brain. We isolate the RNA and we apply the Illumina RNA sequence technology. And after that, we map all the sequences that we have to the genome of the macaque to understand which ones are the differentially expressed genes. From the brain of the macaques that had the viral load in the brain, we found 138 differentially expressed genes that are clustered here for the most enriched pathways. And some of these genes are inflammatory markers and inflammatory mediators, and also some of them are involved in processes of SIV and HIV encephalitis and neurodegeneration. More importantly, we wanted to see which ones are the genes that are expressed in and associated with the Alzheimer's disease pathway, as the HIV associated encephalitis is Alzheimer's disease-like condition. If we take a close look to the markup for the Alzheimer's disease pathway, we see that there is a decrease of activity of the mitochondrial respiratory chain complex, which would lead to mitochondrial dysfunction. And this is one of the triggers it's one of the processes that triggers Alzheimer's pathophysiology. We also see increased expression of calcium voltage gate channels that would more likely allow more cations of calcium in the brain cell, disrupting homeostasis of the calcium and therefore leading to neurodegeneration and neuronal injury. And we also found an increased expression of inflammatory mediators like TNF-alpha and interleukin-1, which also would account for more inflammation and neural injury. In conclusion, we have found that there is a high and diverse viral variation of viral gene flow in the brain that would be a result of multiple introduction of the brain of the virus in the brain. This also leads to subcompartmentalization of the virus, and applying Bayesian file anatomy, what we have found is that the virus evolves from the periphery to the bone marrow and then it adapts in the brain. And studying the gene expression pathways in brain with a detectable viral load, we have seen that the pathways are very similar to the Alzheimer's disease ones, that would include neurodegeneration by mitochondrial dysfunction and also increased inflammatory biomarkers that will mine the neuronal integrity. And this research has been performed by Brittany Rife. There is a PhD candidate in my lab, David Nolan and James Jared Dollar. And these are the collaborators of the Salemi lab. And especially the William lab, that is in Boston College, that is where we have all the macaques, where they provide all the samples that I've been showing you. Thanks. Mm-hmm.
Just as far as the application goes, I think it's really interesting. You can kind of track exactly how these pathologies evolve and what systems they affect. Can you imagine any uses for at least like drug targeting or developing better drugs to treat these conditions?
Well, each cell type has a specific feature, and so if we understand which is the cell type that is involved or contributing more to the viral infection. I cannot, but I guess that people they could understand how to target better that specific cell type. And also, especially in viral rebound, if we understand which one is the reservoir that is more contributing to the reestablishing of the infection after latency, we can target even that to try to eliminate the reservoir in HIV-infected people. That's why it's important to understand where it's coming and how the virus is evolving.
I wasn't sure if I was going to get any introduction, so I went ahead and put the introductory section. How do I get the bottom? I can't see.
Right there, you just pressed it.
I don't know. Oh yeah, sorry. I'm somewhat blind.
Can you put the mic on?
Oh. Yeah, my voice is very loud, so I usually don't, when I lecture, I usually don't put mic on. But I presume this is recorded.
Yeah. Oh, yes.
Okay. Well, I initially thought of presenting on the vaccine mandate, but then I said, well, the audience includes students and faculty member maybe. And how many of you are students? Okay, very good. Well, actually, 30 years ago or more, I worked at University of California at Davis. I was assigned to School of Medicine and to veterinary school. So how could a veterinary faculty, I'm in UF as a veterinary faculty, how could a veterinary faculty work on HIV? Now, I'm working on HIV because of what I did in California. In California, at University of California, Davis, they assigned me to massively produce HIV. And at that time, there were 3 HIV I produced, and that of right over here, Pasteur Institute, Francois Barré-Senouzzi's virus, and that is the French isolate, and also Gallo's NIH isolate, and then I also produced right over here Dr. Levy's San Francisco isolate. The San Francisco isolate is actually the first HIV vaccine in United States, not vaccine, but HIV isolate in the United States. However, the Pasteur group are the ones who really won the trophy of discovery. Dr. Levy said he was only about 6 months late in the discovery of HIV. So why I asked you about students, the reason is I was not a postdoc, but a little bit over postdoc, assistant professor, when I started to work with these individuals. And that's roughly in 1984. I got my NIH grant in 1983, and I basically, and you know my age by now. So I started to work on HIV, massively producing, and my job was to make a diagnostic for Sacramento Valley in California. And we were one of the first to make Western blots for HIV in California, but that was not FDA approved. Okay, so eventually I decided to work with Bio-Rad and made the second FDA approved HIV Western blot. So that's my history. So 1984 I was making, and then in 1986, my job was to look for animal models for AIDS. They asked me to work with monkeys, and I said, well, no, my grant was on cats. Cat AIDS virus, okay, and that was feline leukemia virus. And when I got the grant in '83, I was working on feline leukemia virus, which is the family of AIDS virus, okay. And so I basically decided that since they recruited me to massively produce HIV, NIH was very generous in letting me change my grant into an HIV grant, okay. So doing that, my major thing was to look for animal model. Of course, my feline leukemia vaccine system is what I was interested in. So I said, okay, I'll use the cat model and see if the cats have AIDS. So in 1986, I discovered that feline immunodeficiency virus, or feline AIDS virus, in Petaluma, California. Since then, I have been working on making a vaccine for FIV. It's called FIV. Okay? So what I'd like to talk about today is, I didn't want to talk about how I produced the cat virus vaccine, the FIV vaccine. Instead, I said, what would your audience like to know about? And I think, like myself, I would like to know what is the status of HIV vaccine, and do we still need the vaccine? So the first question we have to address is, can we really, do we need any effective HIV vaccine? And I'm sure a lot of the previous talks were on drugs, AIDS drugs, antiretroviral drugs. So I decided that to present right now why we need, how do I get this? I think you may have seen this slide given by somebody else, another lecturer, maybe not. This is actually the CDC's result in 2013. And basically, the darker blue indicates that they're highly affected counties. And you can see California, which is second in the persons living with diagnosed HIV, has a lot in Southern California. Whereas Florida, we're third in number of people living with HIV. And then New York is number one. So if you look at the totality, there's some areas that are white. Some of the counties suppress data still. Therefore, with the known data, there's roughly 931,000 HIV positive subjects living. However, other estimates are 1.2 million in the United States. Okay? Because the counties don't give you all the data, as well as people don't like to get tested. Okay? Now what you have is a map over there indicating where you're getting a lot of HIV positive people living. Over here in the left right over here, is number of subjects who are diagnosed annually to be HIV positive. Okay? And what you can see, the blue ones are the whole United States. So it's really, if you look at it globally, this is small. There's only about 49,000 people newly diagnosed to be HIV positive. And that is, in 2008, it's 49,000. But if you keep on going down, it's all the way to 2014. The survey shows, and right now we're at basically 44,000 per year. Now, if you look at the gray bar, that's Florida. It's really small in comparison to the whole United States. But if you look at it, it's basically plateaued down right around here, and then it's creeping up. 5,300 subjects are compared to 4,700 in 2012 and 2013. So we have to make sure we don't allow the number of newly infection to pop up in Florida. There's a number of states that are coming out positive for is increasing in HIV. Those are actually in Texas and Florida. New York has decreased. They were the first place where a lot of major HIV epidemic occurred. And then California is still increasing slowly. The number of HIV positive subjects. Now, Puerto Rico, as you can tell, Puerto Rico has about 18,000, but they are not distributed based on county. They only have a total number. So these are part of the United States too. Okay, so District of Columbia, it has probably the highest rate of HIV infection, which is sad. Backwards. Now, do we still need HIV vaccine? So we still have a lot of HIV infections still occurring in the United States as well as world. The world result is down here on the bottom, And if you can see, in 2002, there were roughly about 42 million blue is living with HIV, okay? And then it plateaued down around in 2011, it's down to 34 million, and it's gradually creeping up to 37 million. And this is a little bit worrisome. The drugs are in place, but the drugs for globally came in late. In the United States, the drugs came in, the heart or the combination drugs came in by late 1980s. However, in Africa, was the major country having HIV, the drugs were not that easily available until 2005. It's really a major delay. And there's a number of reasons why it got delayed. One of which is, in U.S., the drugs, annual cost of drug was roughly $10,000 to $20,000. At the peak, it was $25,000 per year. And whereas none of the people in the minority countries have the money to really pay that much. In fact, foundations like Clinton Foundation, I'm not advertising Democrat, but foundations like Clinton Foundation really forced the lowering of the price to the companies, and they are now selling it roughly about, for the low income areas, of $100 a year. Okay, and that is incredible work by these foundations for AIDS. So they have started to treat HIV patients globally starting 2005. And what I want to discuss is also the economic issue, as well as why it is very difficult to make the vaccine. One of which is, you saw that we need the vaccine because ART, antiretroviral therapy, does not cure. In fact, the previous speaker said that ART, and we don't have a vaccine, and there's no cure. Okay? There is no cure because they hide in the reservoir, and as soon as you take the drug out, it pops out. Okay? And that's a problem. So one of the things was to make therapeutic vaccine, or vaccine for therapy, and see if it could increase the immunity. Unfortunately, the AIDS drug does not increase immunity. It doesn't jazz up the immunity against HIV. So that's why they're thinking about therapeutic vaccine. Now, but we still need prophylactic vaccine because you don't want these viruses mutating and recombinating. So you don't want the HIV from subtype B to be combined with subtype C and making more new viruses. When that is already occurring in nature, it already indicates making a vaccine for such a virus is very difficult because it has a higher mutational rate, and it also recombines between HIVs. Now, economically, it's very difficult to have HIV epidemic going on, even with the drugs. The drugs, they cost a lot of money to do the drugs. What I have done is to show you how much money is spent per year. Roughly $10 billion federal government is spending on AIDS drugs and treatment and diagnosis in the United States annually. Of which, how much do we spend for globally? We spend roughly about $6.6 billion to giving money to the world, especially the low-income countries. And that is to stop. You don't want a country giving where you have the source of infection going on in high rate. You don't want those to come to the United States as well. So it's best to prevent it at the major site as well. So there's another issue. And the other issue is, while looking at the United States, we notice the age group that are getting more and more infection, while the other age group is going down. And the age group that is going down is actually our college age group, age 13 to 13 to 24. They're slightly increased compared to the 2008-2014 data shows that it's slightly increasing. Whereas the first group, 25 to 34, is still the same, but this group right over here is increased from 8,800 to roughly 9,700 of newly infection. Okay, so education that we have to do, and that's one of the reasons I'm here to give a talk. I used to do this when I first arrived at the University of Florida, and that is the students and those students who are around that age, you have to be very careful what you do with your life. Now, the other issue is we're really losing a lot of African Americans to HIV infection in the United States. And as you can see, in 2004, the highest number of people infected with HIV, new infection cases, are in the African Americans, followed by white, followed by Hispanics, Asians, and multiple mixed race. So in Florida, it's predominantly heavily homosexual interaction, and more than that, it's actually the prison, the minority black population in prison. And both men having sex as well as heterosexual transmission is a major problem in Florida. Now, I think you saw a similar map from the first speaker. This is actually, the map itself is 2012, but all the other result is roughly 2015. What I wanted to show, much similar to the first speaker, was that subtype B is one of the major ones, and you can see that it's 11% is subtype B. Subtype, and I think you already know the different subtypes, so it basically means groups, and the sequences are more similar in the same group. So we have a fancy way of calling it subtypes. So subtypes B is, you can see C. Now it's color-coded, so majority of the infection is in right over here, South Africa with subtype C, and India. So those in Africa moved to India. That virus moved. Now, what's important about when you're making vaccine, you need to know what subtypes, because the sequence is quite different. And yet, we were hoping that there'll be a consensus sequence for all global HIV sequence. However, it doesn't happen that way. Each subtype is distinctly different. And even more, the difference is even more higher at the envelope region, where immunity targets with antibody immunity. Hopefully, all of you know immunology, because vaccinology is understanding immunology. But so the difficulty of making the vaccine is we have different types of subtypes, A, B, C, D, and then right over here, it has F, G, H, J in there. And then on top of it, you have all the recombinant forms of HIV, okay? And that's why these other codes right over here has all kinds of variation of recombinant forms. But what we want to make first is subtype B vaccine and subtype C vaccine. So majority of the clinical trials are basically located in those kind of regions, those areas for humans. Why is it so difficult? As I showed you, there's so many subtypes. So to make a universal vaccine, you basically would have to put all the subtypes in. But that is not a smart idea. Because how do you select a good virus from a bad virus? Or less pathogenic from non-pathogenic? And secondly, we cannot inactivate a virus and make an HIV vaccine. That's because the fear of having the genome of the virus somehow escape the inactivation process and infecting the humans instead. So in the case of HIV vaccine, there has been a major delay because you have to take only components of the virus. And you can't take the whole virus and kill it and then vaccinate. Whereas I did that. I was able to identify the wimpy virus versus a strong, very pathogenic virus for FIV, feline immunodeficiency virus. And then I basically inactivated the wimpy viruses. And those wimpy viruses are actually better than vaccine virus, okay? But we can't do that in humans. In fact, my second generation FIV vaccine is made out of units, T cell epitopes and B cell epitopes. epitopes are small regions that are immunogenic region, often giving protective or non-protective stimulation. Okay? So why we cannot is, why it's so difficult to make a vaccine? Many subtypes and many recombinants. And HIV mutates, and under mutation, immune pressure, it mutates even more. Also, HIV loves infecting CD4 T cells because the receptor is CD4. Macrophages also have CD4. That's the primary receptor, CD4. And then it also infects dendritic cells. Not as high in level of infection. Now, macrophage and dendritic cells are important for immunity in presenting foreign antigen to the immune system, whereas CD4 T cell enhances immunity of CD8 T cell as well as NK cells, natural killer cells, are some of the typical ones. So it's basically the virus, HIV, loves to infect the important component of the immune system. Very hard to make a vaccine when you need those regions. And in fact, if you stimulate CD4 T cell too much, you enhance infection because that's the prime target for HIV infection. Okay? So there is a problem, difficulty there. Also, another difficulty, making a mucosal immunity vaccine is not that easy. We just got the papillovirus, or we had the genetic vaccine come out for the vaginal tract system. But it is very difficult to make mucosal immune vaccines. And that is for heterosexual men having sex with men. And for IV drug users, we have to have systemic vaccine that works in circulation, in peripheral blood. So basically, the vaccine has to, although the predominant infection is mucosal, you have to make a vaccine for mucosal, followed by systemic. Now, antibody immunity. Majority of the human vaccine, including veterinary vaccine, is based on antibody immunity. That is, you make an antibody to the virus or a foreign organism. And that antibody basically clears like this. Now here, let's say this is a cartoon for HIV, and those blue entity are antibodies. So it binds to the virus and basically neutralizes the virus by not allowing it to infect the cells. And it gets cleared a lot of time by macrophages, okay? And so, but it doesn't infect once you have the neutralizing antibody going on. Now, what happens is, so the vaccine is made out of antibody. So the first two major vaccine trial, it's called phase 3 vaccine trial. Right before commercialization, it's phase 3 clinical vaccine trial, okay? There were 2 phase 3 vaccine trials that occurred. And that was solely based on the envelope of the virus because antibodies react to the surface of the virus. The lollipop-looking entity on the virus are where the antibodies react to. They can't react to the inside of the virus, okay? They don't usually react to the core. It's best to react to the surface of the envelope. And these lollipop-looking entity on the virus are actually are called envelope. Oh, I'm going into a different slide. Well, I can't move it, so here. Basically, the lollipop's right over here. And so the antibody reacts to this, and this lollipop is called envelope protein. Okay? Now, so the 2 phase 3 clinical trials occurred and failed royally. It didn't protect. It was subtype B vaccine-based vaccine. The second one of this phase 3 clinical trial was subtype B and E, okay? And it also didn't work, okay? And a lot of these clinical trial was done in Thailand too, where subtype B and BE is prevalent. Okay? And then right over here, it says, it is not that easy to make broadly neutralizing antibody, anti-HIV antibodies. Neutralizing antibodies that reacts to all the subtypes is very hard to produce. The epitopes are somewhat hidden. The secondly is, okay, the 2 of the major antibody-based vaccine trial failed. So the next question is, how about T-cell vaccine? Merck gave 2 major vaccine trials, and it's called phase 2B trials. 2B is right before phase 3. It gives you a little bit larger number of subjects in the group, and one of which, phase 2B, one set of phase 2B study was done in the United States and Europe and Canada. It's group 1 grouped, and then the second phase was done in South Africa. Okay, so what happened was, and they used to put in a T, so it doesn't have any envelope, but only the other component of HIV. So you're not going to induce the most protected antibody-mediated area, but instead it was GAG protease or GAG pol region, which is polymerase and group-associated antigen. It's the structural protein region of the virus that's inside the envelope, inside the virus, basically inside right here, inside here. So none of the surface entity envelope proteins were not expressed in this vaccine. And this vaccine royally failed. It enhanced infection instead. Okay, and it was used, the vector they used to express these proteins was adenovirus 5. Unfortunately, humans are infected with adenovirus. And some of these cross-reacted. And the company was saying, no, it's not due to the adenovirus. But the problem is adenovirus, this heavier vector, also induces a lot more gamma interferon towards adenovirus. And consequently, gamma interferon is off. When you have it too high, would be bad for vaccine protection. It actually, gamma can enhance infection of HIV, much like my FIV2. So hopefully I'm getting into the last slide. So basically, my interest, I work on FIV vaccinated vaccine. And my commercial vaccine came out in 2002. And that is made out of subtype A and D. And I combined the subtype 2 with the viruses and was luckily able to get protection. My vaccine induces not only antibody immunity, but it also induces T-cell immunity. Okay? And we found out T-cell immunity was protected against subtype B and C because my vaccine is A and D. And the T-cell vaccine, the T-cell immunity was protecting against C and B, whereas A was predominantly by antibody immunity. Okay, my vaccine is A and D. Okay, and the D2 is predominantly more antibody immunity. It's strong. So it doesn't mean that even if you antibody immunity that is neutralizing exists towards A and D more with my vaccine, and doesn't have too much neutralizing antibody towards B or C. That's what it means. And the T-cell immunity is predominantly one major way of protecting against viruses that's not part of the vaccine. Okay? And that's one thing we discovered. And we discovered that T-cell was important. In the SIV field, too, they use a CMG or cytomegalovirus vector and induce it's a little bit unique CD8 immunity, CD8 T-cell immunity that gave 50% protection. Okay? My vaccine usually gives you 68 to 70% protection, especially to subtype A easily. Okay? So, but that is not enough. That is not enough. Because in veterinary, we could give it annual boost. I mean, with this vaccine, maybe every 2 years boost might be the way to go. However, the veterinary community, veterinarians like to make money. So basically, they immunize once a year. I'm not going against veterinarians, but it is one way, and it's actually better immunity that generates too. Now, the HIV vaccine study that there's only one trial that looks promising. Okay, and it had only 1 year to 1.5 year immunity. That was good, and we're quickly going to talk about it. And that's the RD144. It is a combination, like my vaccine, it is a combination of B-cell antibody-inducing system by using the failed ACE, by using the envelope protein that failed in the first phase 3 study. This is subtype B and E envelope. And then priming with GAG-POL envelope of subtype B. In other words, it has GAG-POL means T-cell immunity will get generated if it's going to, if it has any protection. What happened is they had high risk, low risk, medium risk group. If you combine the whole group, they had efficacy of 31.2%. However, if you go after high risk group, it had only 3.7%, which to me is not good. So, and also the duration of immunity was anywhere from 1 year to 1.5 years. And it's not long enough. Okay? Because most vaccine, we want it like 5 to 10 years to maybe 20 years. Because I don't want to be reboosted. Okay?
Was that trial throughout the world, or was it in the U.S.?
This trial was actually done in the United States heavily. And then they did it also in Thailand, I think. So this is a result that looked promising, and it has both T-cell immunity generation as well as B-cell immunity or antibody immunity. Now, what I want to give you a history about vaccine. Vaccinologists was not sitting around now doing clinical trials. They've been doing a lot of clinical trials worldwide. Throughout these blue areas are areas where they did vaccine trials. So let's talk about vaccine quickly. I'm sorry, I'm a little bit long. Phase 1 is dose escalation and safety. Usually has about a subject number of less than about 200, less than 150. And whereas phase 2 has a little bit more number of subjects, and it's expanded safety and immunogenicity analysis. And these are all human clinical trials. Okay? And phase 3 is efficacy trial. You have a lot of subjects, over 3,000. And this study had 16,000 subjects. And they have to have 16,000 subjects to get this kind of efficacy result. Okay? Because these are all contact. You're not injecting it. In animals, we could inject. In humans, we have to be associated. The high-risk groups are men having sex with their partners, or a partner who has HIV who are female and male relationship. So those are, and IV drug users are really high-risk group. So they have to select their population. So what happens globally, these are global clinical trials. There has been 266 official trials. I deleted projected trials already. And 220 are phase 1, 23 is at phase 2 trials, and 20 phase 1 and 2 is a combined trial. And you notice there's only 3 phase 3 trials. This is the other phase 3 trial, and this is the most effective. Now, if you're interested in science, the vector vaccine, majority of these approaches has envelope, induces antibody responses. Okay? And that's probably appropriate because antibody response are much more easier to give long duration of immunity than T-cell immunity. So basically, we still don't have vaccine. Yes, I have a vaccine for my cats because I could kill the virus and then make the vaccine of multiple subtypes and vaccinate the cats and challenge it to know if it's how quickly we could evaluate is another issue. This is a real long-term evaluation system. A lot of these clinical trials are 5 to 7 years trial system. It takes time. So right now we have no vaccine. We think we know how to make vaccine. It will be T-cell and B-cell combined. Any question?
Thank you all for coming, and thanks so much again to our speakers.
Thank
you. If you didn't sign in on the way in, please do sign in on the way out. That's just for us to keep track of how many people are here eating.
So do they use HIV-2 for any vaccine? Is its entry very different?
The entry is identical. The problem is, the issue is, it's not support. Their life is really long, and you don't get the symptoms so long. So, majority of us work on HIV-1 instead of 2.
But if envelope's the same, why does
no, the envelope's not the same. The sequence is different, but they use CD4.
They use CD4.
Yeah. And the chemokine secondary receptors.
But that's what I was asking.
Yeah.
How similar is
Yeah.
similar?
To identification is same, but the sequence is different. There you go.
Thank you.
Got a little bit different. You want to delete? Oh, well, okay. Oh, you're going to delete it, right? Because if not, just delete these two.